Ladies and gentlemen, welcome to the first day of the 2020 webinar series on crystallization, organized by the Working Party on Crystallization of the European Federation of Chemical Engineering. I am Marco Mazzotti, professor at ETH Zurich in Switzerland, and I'm also the chair of the Working Party on Crystallization, and I have the honor and the pleasure to host these webinars on behalf of all my colleagues. The European Federation of Chemical Engineering is a non-profit making association whose objective is to promote cooperation in Europe between professionals, scientific and technical societies in 30 different countries for the general advancement and the development of chemical engineering. The Working Party on Crystallization consists of crystallization scientists and practitioners from academia and industry. It advances the theory and practice of crystallization. It organizes the Triennial International Symposium on Industrial Crystallization, in short, ISIC, as well as other workshops, including training events. And it promotes exchange about standards, protocols, and best practices within the crystallization community. You can access the Working Party on Crystallization through different platforms and social media channels as indicated here. I'm also obliged, before starting, to make clear that the views expressed by speakers or third parties during EFC webinars are those of the speaker and the third party, and not necessarily of the EFC or of the institutions associated with those individuals. After saying this, I would like to start the first day. Today's 2-hour webinar is devoted to crystallization processes. It will be chaired by Béatrice Biscans, who is a research director at CNRS in Toulouse, France. She was the chair of the Working Party before me, and also the chair of the ISIC conference held in Toulouse in 2014. Beatrice, the floor is yours.
Thank you very much, Marco. Good afternoon, ladies and gentlemen. I'm very honored today to introduce this session with keynote speaker. It's with Professor Martha Grover, and she's professor and associate chair in the School of Chemical and Biomolecular Engineering at Georgia Tech. And Professor Grover earned a PhD in mechanical engineering from Caltech and received several awards, an NSF Career Award in 2004, the Outstanding Young Researcher Award from the Computing and Systems Technology Division of the American Institute of Chemical Engineering in 2011. And she is a member of the NSF NASA Center for Chemical Evolution. So Professor Martha Grover is a system engineer using mathematical techniques and computational tools to design, optimize, and control macromolecular organization. You can see here some projects of her group. Her work included phenomena in polymerization and crystallization with applications in polymer organic electronics, pharmaceuticals, and origins of life chemistry. And always experimental and computational approaches are combined, and her overall goal is to use mathematical models for prediction and design of complex chemical systems. So today, Professor Martha Grover is going to speak about a data-centric approach to optimal crystallization control. And now I give you the, Professor Grover, you can speak of your presentation, for your presentation. Thank you very much.
Thank you so much. I'm very glad to have this opportunity to speak today. I appreciate the invitation. Start my video and share my screen. So I will be talking today about our work in optimal crystallization control, and with a particular focus on data coming from process analytical technologies in real time and utilizing that source of data, as well as other data coming from models. And that's what I'll be talking about today. Basically, we'll be focused on going from a disordered state to an ordered crystalline state. So this is not advancing. So I'm going to stop sharing for a moment. Share my screen. And try again. Great. So I'm coming from Georgia Tech, and Georgia Tech is centered in Atlanta, Georgia, which is in the northwest part of the state of Georgia, in the US, right in the city. And in my research group, we look at the control of macromolecular assembly or self-assembly in a variety of applications, as you already heard. And we really focus on the dynamics or kinetics of the process-structure relationship. And this may include covalent as well as non-covalent interactions. So I'll be talking about non-covalent interactions today. And often we're trying to drive the system into a low energy crystalline state, but also sometimes we look at driving the system into a metastable kinetic trap. And so this work was reviewed. This field was reviewed by Richard Bratz's group a few years ago. And I'll be talking today about our work in crystalline control. So as you all know, ordered crystalline states are very desirable for separation and purification, and also in materials processing. In my group, we look at separation as well as materials processing in our crystallization applications. And we're looking at, in particular, how feedback can help us overcome what seems to be sometimes an inherent trade-off between thermodynamics and kinetics. So if we'd like to assemble the system into a crystalline state very rapidly, then we might get defects. If we go slower, we can approach the low energy thermodynamic state. And so with feedback, we can potentially see defects as they're forming and take intervention only when needed, and then potentially get much faster to the ordered crystalline state through feedback. Now, the idea of applying control to crystallization is not a new idea. And Jim Rawlings wrote a paper about this that was quite comprehensive in 1994 on model-based approaches for operating and controlling batch cooling crystallizers. And so that was a really foundational work in the field. In my group, we're working in a number of specific applications of crystallization and control. One of those applications is a collaboration with Ron Rousseau and Andy Bamarius on continuous enzymatic reactions of crystallization for beta-lactams. And so in this case, we're looking at how we can combine an enzymatic reaction in water with a crystallization to trap the intermediate, which is cephalexin or amoxicillin, and how through this intensification, we can have smaller, more efficient and cleaner processes. So I'm not going to talk about that so much today, but this is one of the specific applications that we consider in the group. As mentioned in the introduction, we do theoretical and computational work that coupled closely with experimental implementation. So what I'd like to talk today about is our work in crystallization control. And first, I wanted to review some of the key works in this area with a focus on experimental approaches. And so sometimes open loop control strategies are used with dynamic inputs. Common approach is to use a linear input profile, like in batch cooling crystallization for the temperature, but also there are other approaches used like toggling, so periodic switching between levels in a magnetic field. But also closed loop feedback has been used experimentally in a limited number of cases. So for example, model-free approaches have been used by Zoltan Naj's group, Richard Bratz, and direct nucleation control. Proportional integral derivative controllers, PID controllers, have also been used. And so I cite here a study by my collaborator, Michael Bevan, on feedback control with proportional control for colloidal assembly. And I'll be talking more about that application in the talk. Nonlinear model-based control has been used in a very limited number of experimental cases. Here at the bottom, I cite paper by Ali Mesbah and Zoltan Naj with Kramer and Vandenhoff, where they implemented model predictive control on a crystallizer using a population balance framework. I believe this to be the only experimental implementation that I know of model predictive control in a crystallizer. Here they didn't measure or control crystalline properties, but they did control the concentration. But there are extreme computational challenges to implementing nonlinear model predictive control experimentally in real time. So in our group, we have taken a different approach in which we have used dynamic programming instead, which is an alternative formulation of the optimal control problem, but really the same fundamental equations as in model predictive control, but the policy is calculated offline ahead of time. And so it's very straightforward to implement it in real time with a lookup table. So that's what I'll be talking about today. Model-based approaches. And I'll be talking about two case studies in the group. One is colloidal crystallization of micron scale particles, and the other is small molecule crystallization, and mostly I'll be talking about salt crystallization. So there are a number of challenges for implementing real-time control of self-assembly, and in particular crystallization. There are many atoms or particles, and so there's a large state dimension undergoing stochastic and nonlinear dynamics. Usually we have a limited actuation ability, so we may have a few knobs on our crystallizer, like temperature and evaporation rate, solvent addition, but it's much smaller than the number of particles. And we also have a limited number of sensors for real-time measurement. And so I'll be talking to you about colloidal control, where we actually are watching the assembly under the microscope. But in many cases, we have a smaller number of measures, maybe for some crystalline overall quantities for the crystalline structure, like this C6, that's the local order, and the Psi6, which is the global order. So you can see here some different configurations where we have low order on the left and high order on the right. In the middle, we have high local order, but we have grain boundaries, and so there's a low global order. So the approach I'll be talking about today is something that we have been developing and implementing over the years. And here's one of the earlier papers describing this approach from 2008, cited at the bottom, in which we applied this approach in simulation for a thin film deposition process. But the overall idea is first to select aggregate variables that characterize the overall state of the system and can be measured in real time. So we don't really want to worry about the state estimation problem. And so we want to pick measured variables. So that's really key, but also that can describe the dynamics in a predictive way, in a closed way. So that's, I think, the hardest part of this methodology. Once we have that state, then we can apply machine learning to develop an empirical model of the system dynamics in terms of these state metrics, and then calculate the optimal feedback policy with dynamic programming. And so here is a visual version of the methodology, where we start and we have in the upper left, we have crystalline state. So this is the colloidal assembly problem here. And then we develop some aggregate metrics for the system, and that's then what are plotted on these other axes, these two axes in this cartoon. So we have some aggregate parameters for the system, and then we just observe, we sample the system to see where it evolves. Now, it may be that under a high temperature, the red, it evolves in one area of the state space, and under a low temperature, it evolves in a different area. So we can't always sample the same areas with the same action space or input parameters. But we take those initial samples and then we do clustering. So we do some sort of nonlinear manifold learning, which could simply just be the clustering that I've shown with the boxes on the upper right. But then for control, it's very important that from each state of the system, each discrete state, we know the effect of each action, right? So a next important step is to go in and resample, so that we have both red and blue dots in each box. And so you see that on the lower right. But once we've resampled, now we can decide we can figure out how the system evolves under each action over a fixed time step. And there we construct a Markov state model that just really describes the probabilities of moving between the discrete states under each action. And that provides the input, that model provides the input into our calculation of our optimal policy, which is shown then on the lower left, and just constitutes a lookup table. So we take a measurement, and if we, at that point in time, are in a blue box, then we take, say, the low temperature action in the next time step. If we're in a red box, then we do the high temperature. And then we actually test out the policy in the experiment. And often we go around again because we've actually found some new states. So there's actually another feedback loop around this cycle a few times. So mostly in today's talk, I thought I would talk about colloidal crystallization because that might be something that's less familiar to some of the audience members, but I'll also talk about our small molecule crystallization work using this methodology. But the idea here is that we can assemble these micron-sized particles, and if they have a high crystalline order, they have really interesting optical properties. So you can direct light with them through optoelectronics. You can get negative index of refraction, and you could even make invisibility cloaks, at least theoretically. But if you want to have it really work at the macro scale and become invisible, then you really need to scale up this assembly process and have a very small number of defects. So this is work with Michael Bevan's lab at Johns Hopkins University in chemical engineering. The experiments are all done there. And in the work I'll show today, we have about 300 micron-sized silicon dioxide particles in water. So they're solid particles suspended in water. And we only apply voltage to generate an electric field that moves the particles around. That's our manipulated variable. But we only apply high enough field. We constrain the field so that the particles don't buckle into 3D. So we only have a two-dimensional crystal here. We have reversible assembly by setting our salt concentration to enable reversibility. And we can either push or pull the particles toward the middle, depending on the frequency of the applied voltage. But we have really only one knob on the system that we're going to use today, which is the amplitude of the voltage. And we're going to stick with a fixed attractive frequency. But we have full state feedback by real-time monitoring with optical video microscopy and image processing to extract all the particle locations at real time. And we can actually do this much faster than the dynamics of the system. And so sampling rate is really not a limitation here. So because this is a control talk, I thought we should have a block diagram. Later, I have an equation slide also because it's a control talk. So I thought we should have one equation slide. Here we have our process, the colloidal assembly process. On the right, we have our sensor is the optical video microscopy with image processing. And then I'll be talking about this control policy that we calculate based on the model. And our actuator is the amplitude of the voltage. So let's just watch a movie in real time. So this is this experimental movie, but the color represents the C6 value, the local order of each particle. Does it have 6 nearest neighbors in a hexagonal lattice or not? Lambda is the scaled applied voltage. So now we melt it with a low voltage, and then we push hard, but we get a grain boundary. We also have a void. Okay, so it's kind of working its way out slowly. If we just keep pushing, if we're very, very patient, this one will eventually work its way out. Okay, but it's very slow. So if we're doing manufacturing, that's not very good. So let's try again. Okay, so we'll melt it. We'll push hard. Okay, we have another grain boundary. Now, that time it just assembled perfectly. So this is a highly stochastic system. So using the same operating profile, same voltage profile every time isn't really efficient. We really want to have a tailored approach so that we can heal defects when we see them, but not waste time relaxing the system if it's not necessary. This is a really persistent grain boundary. So that's going to take a really long time to work its way out, So probably we should not just wait. Probably we should take some action. But here we're just doing open loop control with high voltage. We're not doing feedback yet. So those are some of the dynamics of this crystallization system. We do have a detailed simulation for this system. So it's not a population balance model, but it's a many body simulation where we track forces on the particles. The primary assembly is driven by particle field interactions for this applied electric field, dielectrophoresis, but we also have particle-particle interactions. We have random Brownian motion, but overall we can run these simulations that are predictive, and these simulations were developed by Michael Bevan's group prior to my joining the project. So we had that simulation available, which was very useful. We also have this idea that in the group and in the project that we can track particle locations. That can be one coordinate system. And so on the left and in the middle, you can see the field and forces that are applied to an individual particle that would push it to the middle or pull it away. But on the right, you can see a different state representation in terms of this order parameter C6, which is the local order, the local crystallinity. And so we can switch instead to this overall aggregate parameter. And that's something that's already really done in the colloidal field. And then we can see how the free energy of the system changes as a function of C6, but also the applied voltage. So this sort of idea of a reduced state dimension was already natural in the colloidal assembly field. As was also this idea of needing control. And so in this case, there was a paper that was published again by my collaborator, Michael Bevan, in 2012 with a proportional controller applied to the system to really try to drive the system from a melted state to the ordered crystalline state. But what they found was that this controller wasn't really that satisfactory. It didn't always take them. They had steady state error sometimes, and it wasn't really able to deal with anything more than kind of a condensation and pushing on the system, not really subtleties like defects in the crystalline lattice. So when I got involved in the project 5 or 10 years ago, we decided to apply optimal model-based control and to do it using this Markov state model. So in the Markov state model, we really discretize our entire system. We come up with these aggregate order parameters and we discretize them. And we also discretize our time into a discrete time step and our applied voltage, our action space. And then by discretizing everything, we can run simulations of the system and observe how the system moves from one state to another state under each discrete action. So that's really the approach we used here. Even though we had a physically based simulation, we developed this empirical model that was much more compatible with our optimal control approach. And then we were able to form the optimal control problem using Bellman's principle, where we really just say, here is what we would like the system to do at the end. So, and then there's a cost at each stage to taking an action. And so it's a very, very general generic formulation of optimal control. That's also the basis for model predictive control. But here we chose, instead of using model predictive control, we chose to use dynamic programming so we could move all of the calculation offline instead of online. Now, there are some advantages also to doing the online computation. One is that it's more naturally deals with constraints in the system, but that wasn't such a big issue for us. And also, if you have a very high state dimension, then it can be difficult to do dynamic programming. But again, for us, we had a relatively low state dimension. And so we were able to calculate this optimal control policy in the form of a lookup table. So in this case, we used our Brownian dynamic simulation to generate this Markov state model. And we use the Markov state model to calculate this lookup table. So the way the lookup table works is that at each point in time during our assembly process, we calculate from the image the local order and the global order, C6 and Psi6. And then we can look up which color we should do. The red is a low voltage. So if we get into this upper left corner where we have high local order and low global order, that means we have a grain boundary, a serious grain boundary. So then the red says relax the system for the next time step and relax back down again. And then if we're kind of in the orange area, that means, okay, we have maybe a grain boundary on the edge. So, but still we need to relax to move it out. In order to achieve our overall goal, which is integrated over all time, we want to maximize global order, Psi6 squared. So that was our optimal control formulation. But we can also look at some more physical interpretation of that. So we can watch how the system evolves in the upper right on the free energy landscapes for the system. So under each of these different voltages, we can watch how the system evolves and how it jumps between free energy landscapes. So that's kind of just a different way to visualize the system. And we can also look in the lower left at actual particle configurations to see what are the optimal actions under each of these configurations. So it turns out if you're in 4, 5, or 6 on the bottom, even though some are more ordered than others, under all cases, we should apply the highest voltage to assemble quickly. State 1 is actually our desired configuration. But if we're in state 2 or state 3, we have these really defined grain boundaries. And in those cases, we should apply a lower voltage to relax the system to heal the defect. So we applied this optimal policy first.
Martha, can I interrupt you just 1 second? Sorry. Can you maybe try to use the pointer? I think that would be
sure.
helpful to because you're looking at details of your figures and that.
Thank you. I will use the pointer.
Yeah.
I found it. Here it is. So when we applied the optimal policy in the Brownian dynamic simulation, what we found is that if we just pushed hard on the system, about 2/3 of the time it would assemble nicely, but 1/3 of the time we would form a grain boundary. So the blue simulations here are simulations that did not form a defect. So we could push hard and assemble them quickly. But if we have the green simulations, they formed a significant grain boundary, and they did require having some intervention in order to assemble. And so you can see here that even most of the green ones did assemble to high global order by the end. So then we applied it in the experiment. Actually, in this case, it worked similarly, so that was great, but it actually worked even a little bit better. So in this case, in all cases, our control was able to get us to the desired state at the final time. And so we can just see here then in individual trials of the experiment what happened. So first we would melt the system, and then we would push hard to get it to assemble. But here, size 6 did not go up all the way. We have a grain boundary, so we relaxed according to the policy. And we need a couple of times to relax it, but finally it does assemble. Many of the cases did not require intervention, but some of them did. And that's really why we need control, because the system is stochastic, so each trial is different. And okay, so let me go back to turn it off. Okay, so now we can watch some movies. So you can really watch whichever representation you like. You can watch the system jumping between free energy landscapes, or you can watch the actual movie on the bottom. I've turned off my pointer here so I could turn on the movie. But we can see us moving around on the policy map in the upper right. And finally, now we got to our desired crystalline state. Okay, so that's how this optimal control approach worked in colloidal crystallization. I also want to talk about our work with a similar approach to salt crystallization. And this work is funded and motivated by environmental cleanup for the Department of Energy for nuclear waste, particularly that stored at the Hanford site in Washington State. So crystallization is one approach that can be used for separation of the waste. But we're looking at a lab scale system with a waste simulant that consists of sodium nitrate and sodium sulfate in water called duraphskyte. But we have a number of different in situ probes. We have an Optimax here with iControl software for control. So again, we form this optimal control problem. Here, it's a little bit different in that it's a finite time formulation, whereas for the colloidal control, we pose it over infinite time, but we still have a cost function. Let me turn my pointer back on here. We still have a cost function for the distance to our desired state. So this little symbol here is our desired state. And we also have a penalty, a cost of changing the temperature. We'd like to minimize those temperature changes. Now our aggregate variables are the mass of crystals in this batch crystallization, which we measure with a REACT-IR for infrared absorption in the solution phase, and then infer the crystal mass. And then we have total counts measured from FBRM, and this is kind of a surrogate for number of crystals. But we're really just using these empirical measured parameters. We're not worrying so much about how they relate to the fundamental parameters. So this is really an approach driven by the data that can be measured by PAT. So if we just apply a linear cooling temperature, then we get over here where we have a lot of crystals, but maybe we want to have larger crystals, larger crystals. So in that case, we apply our optimal policy, and it drives us by having the temperature go up and down according to these colorful policies, and to achieve our desired state. And here we have a time varying policy, unlike the colloidal control policy. So at 15 minutes, the optimal policy is different than at 115 minutes towards the end of the batch. But here the blues are associated with under saturation and dissolving, whereas the orange and the yellow are associated with a positive, with a supersaturation to drive nucleation and growth. And so now we're going to watch how the system evolves on the optimal policy, starting with no crystals. So let me go back, see if I can get my movie. Good, good. Okay, so we're going to start with no crystals. And so we have supersaturation then. So we're going to drive nucleation. Okay, so now the cord count goes up. We nucleate a lot of crystals. We back off on our supersaturation to try to achieve this target over here. We pass it a little bit. System is stochastic, but we turn around and then we can come back here and park on top of our target. Okay, so just wanted to summarize then these two examples and to compare them. Okay, so we have we've talked about two case studies, the colloidal crystallization and the salt crystallization. So in the case of the colloidal crystallization, we have these micron-sized solid particles, and we're pushing them together with dielectrophoresis to form this crystal of particles, of micron-sized particles. And we characterize that system in terms of two aggregate parameters, the local order, C6, and the global order, Psi6. Now, in the case of the salt crystallization, we're actually nucleating and growing crystals in solution. And in that case, we're going to characterize the system in terms of the total mass of crystals, and actually not necessarily the total crystal number, but the total cord count, which is can also represent to some extent the total number of crystals. Well, it's really total cord count. But so in both of these cases, we chose two order parameters to describe the system. Now, in many cases, you might need more, and probably we also needed more. But the wonderful thing about feedback is that it can help correct for some model inadequacies. So, but not all. So in the case of the colloidal crystallization, we tried some manifold learning techniques that were not super successful, actually, to identify our order parameters. And then we picked, so we also then just tried some common order parameters. And these were not the first two that we picked. Some combinations we picked, like radius of gyration and local order, were not really sufficient to describe the future evolution of those parameters. So we did iterate through some different choices before we settled on these. So ideally, you know, these would be automatically generated to be a full state representation. But in our case, we really picked based on physical intuition order parameters for both cases that we thought could just be closed in terms of the dynamic evolution, and had some iteration until we converged on these that seemed to work pretty well together with our feedback. But I do think the selection of aggregate variables is the key challenge in this field, and there's more work to be done in that field. But once we had picked those composite parameters, then we could move on and learn the dynamics of the system. In the colloidal crystallization case, we had significant stochastic behavior. So every time that we did we ran the experiment, sometimes it formed a grain boundary 1/3 of the time. 2/3 of the time, it did not form a grain boundary under the high voltage. So it was stochastic. So we learned a stochastic model. Okay, so we discretized the state space, the time, and the action space, and then we ran a lot of simulations from our Brownian dynamics simulation, and we constructed estimates of the probability of transitioning from one state to another state over a particular action. Okay, so we built these probability matrices for this Markov state model. Now, in the case of the salt crystallization, we actually did not have a fundamental model. So we had not built a population balance model for this system, and we decided to learn the model directly from the data. So again, we did a lot of discretization, but we built a deterministic model, kernel-based model, from our experimental data, we didn't observe so much stochastic behavior except for the primary nucleation. But basically, to get things going, we would start with no crystals. At high supersaturation, we would just wait until something happened. And so that was the primary stochastic phenomenon. And so in any case, here we built a deterministic model from our data. But we could have built the transition probability model, but we didn't take that approach in this case. But once we had this dynamic model, we could then, in a pretty straightforward way, calculate our optimal control policy. And for the colloidal crystallization, we chose to use an infinite horizon formulation. For the salt crystallization, we really wanted to get to a desired point at the end of our batch. And so we decided to use a finite time formulation. But in both cases, we have these colorful lookup tables that tell us what is the best action given that we're in a particular state at a particular time. And our input for the colloidal control was the amplitude of the voltage applied to the quadrupole electrode. For the case of the salt crystallization, we actually used the supersaturation in our calculation as the input. So we didn't use the temperature. Now the temperature was used to drive to the supersaturation to a set point, and we had a local kind of a cascade control structure and a PID controller to control supersaturation as a function of temperature. But for our optimal policy, we chose to use supersaturation as our input because it was a more fundamental driving force, and we had a good physical model for the relationship between the temperature and the supersaturation. And so this actually ended up being kind of a hybrid modeling approach where we used limited data or information that we had about the system in order to inform our overall empirical learned model. So I'd also like to talk a bit about some work that we're excited about going forward. So going forward with this approach to optimal control, there are a couple of things that we're doing. So we're looking at higher dimensional spaces, not just a two-dimensional feature space, but a higher dimensional space. And we're also looking at bringing in population balance modeling into the approach. So if you remember, for the colloidal control problem, we had a detailed model and simulation. We had the Brownian dynamics simulation. Now we still learned an empirical Markov state model from that detailed simulation, but it's very useful to have a detailed simulation instead of relying on just
Okay, the speaker has had to disconnect and she's reconnecting. So I apologize. We're waiting for Martha Grover to join the meeting again. So we need to wait a little longer. I apologize again. Let's wait a few more minutes. Okay, there seems to be difficulties for Martha Grover to join the webinar again. So let's wait one more minute. In case she doesn't manage, I suggest that we move on with the second talk. And when she joins again, we can continue with her, with the final part of her talk. So I suggest that you, Beatrice, and the next speaker get
Yes.
kind of ready. We wait still 1 minute, and if Martha doesn't
Okay.
join us, I would move on to the next talk.
Okay, so we wait 1 minute.
Yes, and Ashwin will tell us when we should
Okay.
move on. Thank you. Yeah, Beatrice, I
Yeah.
suggest we move on. And
Okay.
then, thank you very much.
So if you are ready.
Now
so
Ashwin
now
is telling me that she's back. Just 1 second. One second. I don't see her, but yes, she's back. Okay. Go on, Beatrice.
Okay, you can go on. You can share your screen.
So I'm back.
Okay, perfect.
I'm not sure what happened or when exactly.
Yes, I had the same. Okay, thank you very much for. So now you can share your screen again and go on.
Okay. So I believe I know where things cut out.
Yeah, you were starting to talk about your next projects.
Yes. Okay. So this is a collaboration with the Federal University of Rio de Janeiro and a visiting graduate student, Marcellus Moraes, who was visiting our group for 6 months, working advised by Mauricio Souza and Argamiro Secchi. And we were looking at shape control, so mass, count, and shape, and using image processing as the real-time sensor. So this is their experiment. at the Federal University of Rio de Janeiro, where they have an external sampling loop to do the image processing. And using this data and experiment to build a population balance model, Marcellus was then able to calculate the optimal feedback policy to achieve different points in this mass length space to get different mass and shape. So you can see for some points, a simple
we
pooling.
We don't see your screen. Sorry.
You haven't share. You have to share your screen again.
Yes, I do.
I think that.
Yes, I do. Yes, I do. Thank you. Here's a picture of the experimental set up with the external sampling loop. And so here's the feedback policy. Get the laser pointer here. So for some points, some desired points, we're able to just cool to achieve that point, but under others, it requires going up and down in terms of supersaturation. So actually going undersaturated to achieve the desired mass and shape at the final time. And so this work now is ongoing to implement it in experiment, to validate the approach in experiment. So with that, I'd like to conclude. I think that it is possible to do model-based optimal control at the micron and nano scale, particularly for crystallization. And that selection of the reduced order state is still an ongoing challenge, as well as kind of accuracy of the model and robustness to modeling errors. But it's exciting to me that the technology is continuing to advance for imaging, as well as in process analytical technologies, and that by having a lot of microscopy, it might not be practical for manufacturing, but it can give us insights into what we really need to measure, which might just be a few order parameters. So I'd like to thank my collaborators, Michael Bevan on the colloidal control work, Ron Russo and Yoshi Kawajiri on the crystallization control. Yujun Xu was a postdoc who did a lot of the work, early work on the colloidal assembly control and defining the optimal control approach. Xuan Tong was a graduate student who did much of that work and is now just started this fall. He's an assistant professor at Louisiana State University in chemical engineering. And Dan Griffin, who did much of the work on the data-driven small molecule crystallization and is now at Amgen. And my research group who's carrying on with the work. And the work was funded by the National Science Foundation, the Department of Energy through a fellowship, and also Industrial Consortium, CRESP, and ongoing work with the Food and Drug Administration. And here are a few publications if you'd like to read more, a couple of the earlier works on the colloidal and salt crystallization control, and a recent overview in the Journal of Process Control, and then also a recent paper on our work in the beta-lactam enzymatic reactive crystallization. So thank you, and I would be happy to take any questions.
Thank you very much, Marta, for this very interesting presentation. Now we can have some questions. The first one is coming from Marco Mazzotti. A question on your slide 28, maybe, how did you select the target state in the diagram on the top right, particularly the value of N?
Very good. Okay. So actually, we tried different target states because we wanted to see where we could go. But the angle in this space is really related to the size of the crystal. So for a given mass, now if we have more total counts, then we probably have more crystals. But FBRM is not so easy to interpret. So it's really what we did here is, instead of saying we want to have a certain concentration of crystals, what is the corresponding total count? We learned the model from the total counts, and we did offline calibration to get back to number of crystals. And so we were able actually to develop a linear correlation between total counts and total number of crystals for representative crystals that we were making here. But so we really did that offline calibration to get back to number of crystals and size and crystal size. So we did have some idea about that, but we kind of led with the PAT measured numbers in terms of our modeling and our control, and then we calibrated back to get to physical quantities that we could also, model with population balance and things like that.
Okay. So it's a complementary equation. So are you in the next slide? For Marco, how can you reduce N using which operating variable? So you have both growth and dissolution. Mm-hmm. And how do you know that your target point is reachable in that case?
Oh, how do we know it's reachable? It might not be reachable. So optimal control is giving you best, but best is not always good. And not all points are reachable. So what we did in this approach is we chose some different targets, and then we looked to see, were they reachable, you know, given our optimal policy. But there's no guarantee that all points would be reachable. Now here, blue means that we should apply undersaturation to dissolve, you know, which makes sense. I think our chord counts would be reduced to move us back in this direction. Over here, you know, then we should go forward. And some of this blue here is really noise from not having enough samples from our data, you know, so probably this speckling is not, we didn't actually sample the space up here so much, you know. But here we want to drive growth and nucleation to move right, and we want to move back this way left with undersaturation. But we did also try different final times for our policy, because it might be that a point is reachable in a longer time, but not in a shorter time. So we just did some trial and error there, trying different final times to see, could we get there or not? So kind of the concept of reachability, but we tried different times, batch times.
Okay, and Ashwin is asking for, can you tell more about the crystal that you're going to want to control the shape? How do you measure the size and shape of these crystals?
So we are doing real-time image processing. Okay. So that's the shape is, and so it's a projection, it's a 2D projection. Okay, but because of the shape of the crystals, let's see. I was trying to close my camera with my laser pointer, but that's okay. So because of the shape of the crystals over here, we know that two of the axes are similar, and so we can infer the L1 and L2 from the images. Average, okay. So it's the mean.
It's
mean
a two-dimensional measurements.
Two-dimensional.
Two-dimensional. Okay. So maybe I can add one question about the scale of the measurements you perform particularly in colloidal crystallization. So you had to go to local parameters such as local water or global water, and in such systems, can we imagine to exploit other measurement, for example, with scattering or diffusion measurement or such local, very local measurement? Can we try to export your method to other type of local parameters obtained by other measurements?
Sure. The selection of, in our approach for the selection of the aggregate variables, they should be key quantities of interest that we want to control. They should be measurable, right? So often, you know, you just lead with the measurements that you have, right? So you pose the aggregate parameters in terms of measurements that you have or are interested in. But they also need to be complete in a dynamic sense, so that if we know the current values and the action, we can predict the future values. So all of those kind of requirements need to come together in our aggregate state variable for this to work. But certainly it could be anything. It could be anything that you can measure.
Okay, thank you very much. Very interesting. I think that if we have no more question, we have to move to the next speaker. And thank you very much for this very interesting presentation. And I think that we have more. We are going to read your papers now to have more information. Thank you very much.
Thank you,
Martha. So now I introduce the next speaker. So please, can you change your slides? And the next speaker is Jonathan Gaensch he is a doctoral student of the group of Andreas Seden-Morgenstern in Max Planck Institute of Dynamic Complex Technical Systems in Magdeburg in Germany. So you're going to explain us your project on continuous racemate resolution via coupled fluidized bed crystallization. I leave you the...
Yeah, thank you for this kind introduction. So welcome everybody to my talk. So my talk is about continuous separation of enantiomers. And I would like to show you today the benefits and the advantage of using fluidized bed crystallizers for this purpose. And here at the beginning, I would like to mention that this work is part of a larger collaboration between the Max Planck Institute Magdeburg, where I'm from, where we are working on the experimental investigation, the Ruipeng, where our colleagues developed a reduced process model, and the Otto von Guericke University Magdeburg, where our colleagues using detailed CFD-DM simulation to study all these hydrodynamic effects of this process. So since I'm from the Max Planck Institute, my talk will focus today on experimental results, but at the end, I also would like to compare some of these results with the reduced process model. So let's start. So first, first, why do we have to separate enantiomers? So we have to separate these pair of molecules which just differ in their spatial arrangement, like here our D-asparagine and L-asparagine, because they have different effects in chiral environments like all living organisms. And to separate now these both very similar molecules continuously, we are applying the technique preferential crystallization in these conical shaped tubular crystallizers. So how is it working? So we are starting from a solution with racemic composition. That means that our L-asparagine and D-asparagine have equal amount and equal concentration. And we are pumping this racemic solution continuously from the bottom through our crystallizer. By cooling, we create some supersaturation. And then we seed the process with crystals of just one enantiomer, our target enantiomer, for example, now the L-asparagine. So then these L-asparagine crystals will start to grow and thus selectively just remove our target enantiomer from the feeding solution. So what are the advantages now of using these tubular crystallizers? So first we have this conical shape, and due to this conical shape, we have a varying fluid velocity over the crystallizer height. And this results in a size classifying effect. That means that our larger particles will be located here at the bottom of the crystallizer, and smaller particles will be located here at the top of the crystallizer. So we can expect that our product, which we can withdraw here as a suspension, has somehow a narrow product crystal size distribution, and also that this distribution is also adjustable via the volumetric flow rate. Further, we have a very narrow and defined residence time distribution of our liquid phase, which is important since we have to avoid any nucleation of our counter enantiomer. But even if our counter enantiomer would start to nucleate now, then we could expect that due to the size classifying effect, these tiny crystals will get discharged at the top of the crystallizer. So these both effects promise that these processes a bit more robust than, for example, stir tank crystallizers. And this allows us to be a bit more aggressive, so we can set higher supersaturation to achieve higher productivities. And what means now higher productivities? Just to give a benchmark. So there is a nice paper from Kolges and Vetter, which gives an overview about reported productivities for the preferential crystallization. And all these reported values are below 20 grams per liter per hour. So we are expecting to get higher productivities than these 20 grams per liter per hour. But we have to admit that this process is also very complex, since we are coupling now crystallization kinetic with all these hydrodynamic effects and also continuous seeding strategy. So also the operating window has some further limitations. For a couple of enantiomers, we already could prove the successful racemate resolution. So for the amino acid threonine, for a very challenging API called guaifenesin, and also for our amino acid asparagine, which crystallizes as a monohydrate. And this asparagine we choose as our model compound for our parameter study, because first it's a conglomerate, which is a requirement for the preferential crystallization, but also because of its beneficial properties in terms of the crystal shape. So we have very nice compact asparagine crystals, which also have a fast crystal growth and a solid density, which differs significantly from the liquid phase density. Our pilot line consists now in 2 of these tubular crystallizers because we want to separate both enantiomers from each other at the same time. These crystallizers are coupled via a feed tank, which contains our racemic feed solution and some racemic excess. And the continuous seeding is realized using this milling bypass. So how it's working, our large enantiopure crystals are withdrawn here, sent to the high-speed disperser, our mills, where they get crushed. And the enantiopure fragments we are sending back to the process as our new seed material. So regarding the product withdrawal, so due to the scale of our crystallizers, a continuous product withdrawal is not practical. That's why we are using these 2 photosensors to realize a periodic product withdrawal. And the operating parameters of this process are now first our crystallization temperature, then the supersaturation, which we can set via the saturation temperature here in the feed tank, then the volumetric flow rate of our feeding solution, so which we can measure and control here with our colorimeter flow, Cori flows, and also our product withdrawal strategy. So when and how often do we withdraw our product here at this product outlet? To evaluate now this continuous process with this periodic product withdrawal, we are using the periodically harvested product crystals. So here you can see an image of our product crystals, and then we do some analysis. So we are using HPLC and XRPD to check the purity and also the crystal lattice, and we're also using CIFA analysis. And then we are defining some characteristic numbers of the process. So we are using the mass of our product and time window between 2 withdrawals and also the scale of the crystallizer to define a productivity and the yield. And our product crystal size distribution is represented by the mean crystal size and its respective standard deviation. And using now these 4 characteristic numbers, we could already prove the feasibility of the continuous operation in preliminary runs. So here you could see now the mean product crystal size, the standard deviation, the productivity, and the product purity over the process time. And what we could observe is first that we have a successful resolution of our asparagine. So all these product purities are over 98%. And all these numbers become constant after more or less 2 hours process time. So we could prove that this process reaches a cyclic steady state, even with this periodic product withdrawal. By repeating now these experiments, we also could further prove that these results are reproducible. So here you can see now the product crystal size distribution and the productivities for 2 experiments. And as you can see, there are no significant differences. Now I would like to show some selected results of our parameter study which we performed. So here first regarding the supersaturation. Now here are productivities for 4 different supersaturation. And what we can observe is first that the productivity increased with the supersaturation as expected. But what we further could observe is that our operating window is limited by a certain maximum supersaturation where our counter enantiomer starts to nucleate and contaminate our product. Further, we could observe that our operating window is also limited by a certain minimal supersaturation where our crystal bed stops growing, and as a consequence, our productivity becomes 0. Regarding now the volumetric flow rate, so here you can see now 3 product crystal size distribution for 3 different volumetric flow rates, 10, 12, and 14 liter per hour. And we observe higher volumetric flow rates causes larger product crystals, as expected. But what we could prove with these experiments is first that our product crystal size distribution are quite narrow and we have a very less amount of fines. And also we could see that all these product crystal size distributions differ significantly from each other. So we could prove with these experiments the adjustable and also narrow product crystal size distribution of this continuous process. Regarding the periodic product withdrawal strategy, here we could observe that the height of the crystal bed has an unexpected strong influence on the process performance. So what did we do? So starting from our reference strategy, we changed the position of these photosensors. And this results, and finally, so we're getting 4 different stationary operating points, which mainly differs regarding the maximum height of this crystal bed. So the volume of withdrawn product suspension was constant for all these operating points. We are just changing the height of the crystal bed. And here we could see or could observe that first our product crystal size increases with this crystal bed height, which is nice to know. But which is more important and was also more surprising is the significant increase of the productivity and the yield. So starting now from our reference strategy, 30 to 40 grams per liter per hour, we were able to double or even triple our productivity and yield just by changing the position of these photosensors. And here I want to highlight once the quantity. So even without optimization, we already could observe now productivities up to 90 grams per liter per hour, which is far above the before mentioned benchmark of 20 grams per liter per hour. So why I just said now 90 grams per liter per hour? Because here we also could find for this last operating point that after short operation time, the counter enantiomer starts nucleate. So also here we could observe some limitations of the operating window. Before I conclude now my talk, I would like to use these results to make a comparison with the reduced process model. So here you can see again the productivity and the mean product crystal size for these 4 operating points. And the reduced process model was developed by our colleagues from the TH Bingen. All the assumptions and equation can be found in the paper from Professor Michael Mangold. And yeah, this model based on the population Belling's equation, which describes a number density function. And this depends on a single property coordinate, the crystal size, and a single space coordinate, which refers to the height of the crystallizer, and the process time. It's only considered crystal growth, and the crystal velocity can be described by the model of Richard Nensaki. For the results I want to show you soon, there have been some refinements. So now our colleagues also implement this periodic product withdrawal and also an energy balance to the model. And further now the model distinguishes between these both enantiomer, our D enantiomer and L enantiomer. So in the future, we also could have a look on purity aspects. And here you can see now the results as black dots, the calculated results from the reduced process model. And what we can see is that, that all these calculated trends of the mean product crystal size and also the productivity and the yield can be confirmed by our experiments and also show a rather good quantitative agreement. And here I would like to mention that also no parameter of this reduced process model has been fitted to these fluidized bed experiments. So let me summarize. So I hope I could show you today that using this fluidized bed crystallization enables the successful continuous racemate resolution of our asparagine with high purity and remarkable productivity for both enantiomers at the same time. And even if our process is not 100% continuous, but even with this periodic product withdrawal, the process reaches a cyclic steady state and the results are also reproducible. Then I could show you some results of a comprehensive experimental parameter study, which was directed to study the influences of the operating parameters, but where we also could find some limitations of the operating window and we could prove this adjustable and narrow product crystal size distribution. And I just show you that there has been already a reduced process model developed, which has a very good quantitative agreement with our experimental results. So what's next? So soon we will finalize a paper together with our colleagues from the TH Bing and the Otto von Guericke Universität, which focus on the optimization potential of this crystallizer geometry, which I was not talking today about. And then we also would like to use the reduced process model for the optimization of the whole process. And in the future, we are also interested in using this separation technique for other isomers, not only enantiomers. And yeah, then I'm at the end of my talk, and I thank you for your kind attention, and yeah, stay ready for your questions.
Thank you very much. Very interesting. So the presentation is open for questions. So maybe the first question asked by Ashwin, I selected, is this strange behavior? What is the physical reason? Why the bed 8 was such a big effect? And with this question, maybe I can add one of mine. Can you tell us some observation of the hydrodynamics of your fluidized bed, and how is the flow inside, because you have a conical shape, and so the velocity maybe is different from one point to another. So Z8 is correlated to the hydrodynamics of the fluidized bed. So can you give some explanation or some observation?
Okay, then I start with the first question. So related to this influence of the crystal bed height. So we are attributing this significant decrease of the productivity because now we have a large population of product crystals or of crystals. So we have now much more crystals in our fluidized bed, which are growing and moving towards our milling unit. So, and if we are sending now more crystals per time to our milling unit, we are creating more seed crystals. And if we have more seed crystals, the whole process will accelerate. So that's how we can explain the significant increase of the productivity and also of the yield. And yeah, about the hydrodynamics. So maybe I go back. So we have the first fluidized bed here, and what we can see from the results is that the crystal, so that the velocity of the liquid phase is also changing, and this also depends on the conditions and the withdrawal strategy. So why we have now here larger product crystals? Because the bed gets compressed. So we decrease the void fraction here. So the velocity of the liquid phase are faster, and that's why our force balance is shifted towards larger product or larger crystals.
Okay.
Is this a good explanation?
Okay, and Ashwin is asking if, do you think that this technology can be scaled
up?
Right. Yes.
I mean, yes, I think yes. I think then one problem is the cooling. So the cooling along the crystallizer wall, because if we scale up, we change the ratio of the volume to our heat transfer surface. So this might be a problem because then we need a larger temperature gradient of our coolant, and this could causes nucleation, secondary nucleation at the crystallizer wall. So this is an issue. So yeah, that's a problem of the scale up, I would say, a big problem.
Okay, and maybe the last question from Francesca Breviglieri from Zurich ETH. How do you choose the amount of suspension to be withdrawn?
So first, how can we adjust this volume? So we have these two light beams. Now I just show here one light beam, but we have two light beams, and by the distance of the light beams, actually, we can set the volume which we are withdrawing. And we also did some experimentation. So, so how does the process change if we now if we are now withdrawing more volume? But actually, it's just unbeneficial effects of the process, because if we are withdrawing now, for example, a huge volume in short time, then our liquid phase also will decrease, and we can have some nucleation here at the top of the crystallizer, because here starts some evaporation because the decreasing level of the liquid phase. So, and we try to make this volume small as possible, because then we are more towards the continuous operating.
Okay, thank you very much. We have to stop here, and thank you again for your presentation, very clear presentation. We move on the next presentation now. Okay. So it is given by Ruipeng Chen, who is a postgraduate student at the Hong Kong University of Science and Technology, and is going to speak about integrated continuous crystallization and spray drying of insulin. You can begin if you are ready.
Okay, so can you guys see slides? Okay, thank you for the introduction. And first of all, I would like to thank the organizer for inviting me as a speaker. I'm Ruipeng from Hong Kong University of Science and Technology, and my presentation today is basically about the integrated process, which combines continuous crystallization with spray drying to produce insulin crystals for pulmonary delivery. So the story is related to diabetes. Diabetes is a common disease worldwide, and the insulin remains the most important treatment to diabetes. So, and insulin is typically administered by subcutaneous injection, which has formulations of different duration. But the problem is subcutaneous injection has low patient compliance and can be dangerous if administered incorrectly. So a lot of efforts have been made to develop alternative non-invasive routes. And so far, there's only one product available on the market, of which is pulmonary delivery. So for this product, insulin is packed in cartridge, and to inhale the insulin, one just put the cartridge into the inhaler and then inhale deeply through the inhaler. This product is limited to rapid-acting Olin, which has a duration of less than 2 hours. And a probable reason is the amorphous insulin it contains has fast dissolution properties. And in contrast, insulin in the crystalline form has many benefits, like prolonged blood sugar control, which is ascribed to the sustained release of the insulin monomer. Sorry, I changed to the laser. Sustained release of the insulin monomer from the crystal matrix. Other advantages are like higher stability and bioavailability expert. So this suggests it would be attractive if we can produce insulin crystals for pulmonary delivery, which requires an aerodynamic size range of 1 to 5 micrometers. And since insulin was first crystallized in the 1990s, it has been studied for various purposes. For example, to get high quality single crystal X-ray data by growing large crystals. And here we want to make general conclusions regarding insulin crystallization. So first, a careful pH control is needed because the solubility of insulin is sensitive to the pH. And as reported in this paper, who use carbon dioxide as the acidifying agent to adjust pH and then create supersaturation. The second, the crystallization of insulin or proteins are very sensitive to shear, which as reported in this paper, who studied the effect of shear on oscillator flow. And third is so far producing a dry powder of insulin crystals in a single step without any particle correction steps is still challenging, and the crystallization of insulin has been reported only in the batch mode. In recent years, there's a growing trend towards continuous manufacturing, in the biopharmaceutical industry due to advantage like consistent product quality and tighter equipment integration. For continuous protein crystallization, it has been studied in tubular reactor, the MSMPR mode, oscillatory flow, and the membrane process. For continuous tubular crystallization operated in slug flow, it has in which the slugs, the liquids are separated typically by the airs, and therefore each slug can serve as a mini batch, which has good mixing and narrow residence time distribution, hence a good, potentially a narrow crystal size distribution, a narrow CSD. Apart from that, it also has typically fast startup and low shear. This is important for protein crystallization because proteins are considered fragile under shear. But the shortcomings are like long reactance time are typically impractical due to the limited tubing length, and also fouling is possibly to occur. It has also been reported that a different attainable size range can be achieved for continuous process compared to a batch one. So actually, in the previous work of our group, we have used the segmented flow crystallization to produce the inhalable crystals of two model compounds. And the tubular reactor was integrated with the spray dryer. Spray drying is a process which can produce a dry powder from a slurry or solution within seconds. And the reason for the integration is due to the need for the fast solvent removal to mitigate aggregation oswald ripening, which is likely to occur in a suspension. So oswald ripening, in brief, is a phenomenon which where the coarse particles could grow at the expense of the fine particles redissolving, and aggregation and oswald ripening. These two phenomena can affect the bioavailability and reduce the stability of the product samples. So in this work, it was also shown a high fine particle fraction from spray drying was obtained compared to the conventional vacuum drying method due to reduced agglomeration. So this work, in combination with other information I presented, inspired us to design the current project, in which we aim at develop and characterize integrated process which can produce insulin crystals for pulmonary delivery in a single step. So for standalone crystallization, we consider the insulin recovery and particle size as the important quality attributes. And for the integrated process, it is the process yield and aerosol performance. Okay, so let's first begin with the solubility data in which it is measured for the typical crystallization range for insulin in the pH range of 5.8 and 7.2. So as we can see in this figure, there's huge difference in the insulin solubility at different pHs. So this suggests high subsaturation can be generated if we apply pH shift in a tubular reactor. So now is the experimental setup. So basically it consists of a tubular reactor and a spray dryer. For the tubular reactor, there are two phase solutions. One is the insulin solution, in which insulin is dissolved in the citrate buffer with acetone at higher pH by heating at 50 degrees C for 10 minutes and then cool to room temperature. For the precipitant solution, it was citrate buffer at lower pH with acetone and zinc sulfate because zinc is the essential component of the insulin crystals. And then a peristaltic pump was used to transfer the two solutions into the tubing via the first steam mixer. And the nitrogen was incorporated into the liquid using an airflow controller via a second mixer, which is placed around 3 centimeters downstream of the first steam mixer. So for the tubular reactor, it was composed of 2 pieces of tubing. Each is 40 meters long, and it was immersed in a thermostatic bus for temperature control. And therefore, the supersaturation can be generated by both cooling and the pH shift. And at the outlet of the crystallizer, we have a buffer vessel. And for the standalone crystallization process, this can be used for sample taking and also to monitor the startup dynamics of the tubular crystallization. For the integrated process, the product slurry in the vessel is continuously transferred into the spray dryer using another peristaltic pump, which is operated at the same flow rate as the liquid flow rate in the tubular crystallizer. So for the spray drying, it was performed at the fixed conditions, and this condition resulted in an outlet temperature for all conditions of around 85 degrees C, which is below the threshold of 120 degrees C to avoid the insulin degradation. For the so the variables for standalone crystallization are pH, which is in the range of 5.2 to 5.8, 7.2 the initial concentration. So this to determine the supersaturation, and the liquid and the gas flow rate, and also the residence time. And the temperature are fixed at 10 degrees C. So we can first look at the startup dynamics of the tubular crystallization. This is monitored by characterizing the insulin recovery and the CSD at crystallized outlets at different times. So here, the insulin recovery is defined like this, in which the C0 is the initial concentration and C is the measured concentration as the crystallized outlet. The elapsed time is time passed since the first product actually crystallized, and two conditions were selected, which differs only in the initial concentration. And triplicate experiments were done of each condition. And as we can see in this figure, so both condition has a relatively low recovery at the very beginning, but soon in around 5 minutes, both conditions reach a plateau and was approximately constant for the remaining operating time. And then we can look at the monitor the CSD at the crystallized outlet for these two conditions. So the trend is similar to the insulin recovery. For both conditions, CSD were on the right at the very beginning, and then start to shift to the left. And condition A and condition D reached an approximate plateau in around 10 and 15 minutes, which is slightly lower than compared to the insulin recovery. The condition A with a higher initial concentration reached the plateau faster compared to the condition D. The reasons why there's shift in the CSD, we think this can be possibly explained by the presence of the liquid foam in the segmented flow, which is determined by flow velocity and the viscosity and other parameters. So small particles with a size less than the film thickness, it is possible to travel between slugs, and therefore the slugs that were formed initially, there's a reduced number of particles, and therefore the CSD were on the right. We also monitored the insulin recovery for other operated conditions, and it was found that for most of them, they reach a plateau when around 12 ml of the product slurry has exited the crystallizer. This is 20 minutes for condition A and D. And therefore, this time point was used as a representative point to compare the process performance. So first, we can look at the effect of pH on the particle morphology, and two pH were tested. One is pH 5.8, which has a very high saturation. The other is pH 6.1. And for most of the particles obtained at the pH 6.1, they appear the typical crystalline shape of insulin crystals, the rhombohedral shape. This is more clear if we look at the microscope images with a higher magnification. While for the particles obtained at the lower pH, which has a very high saturation, a large number of the particles appeared amorphous, and also the agglomeration can be observed. So the particle morphology is also consistent if we look at them on the SEM, as you can see in these two images. We can also, so then we can look at the effect of pH on the insulin recovery and the mean particle size at the initial concentration of 2.4 and different pH values. So for the highest recovery was obtained at lowest pH, which has the highest saturation. This is because the highest supersaturation has the fastest nucleation rate, and therefore the supersaturation is assumed more rapidly. And the larger particle size was obtained at pH 5.8 compared to the pH 6.1, and we think this is because of the agglomeration as observed on the both microscope and SEM. Then we decided to conduct the following experiments at the pH 6.1 because this one has a high recovery and also a small particle size. So we can look at the effect of initial concentration, which perform the pH 6.1 and different initial concentration. So the trend is similar to the pH. So at highest initial concentration, we achieved the highest recovery, which is over 90%. And the particle size was also the smallest. But the particle size obtained at the pH 5 at the initial concentration of 1.2 is also small. We think this is because due to the lack of drawing force for crystal growth at a low subsaturation. And considering this, we decided to perform the following experiments at the initial concentration of 2.4. And then we studied the effect of flow rate and the residence time on these two quality attributes. Now there's a lot of information in these slides. Just want to focus on, well, just want to point to a few general conclusions. You can see for all these conditions operated in the same pH and initial concentration, but with different flow rate and residence time, they all achieve a recovery of over 85 and even 90%. And for the mean particle size, the range of 6 to 10, which is very close to the desired size range. And of course, the measure, the mean particle size cannot be directly translated to the aerodynamic size range, but we think this is very close to the size that we want. So then we decided to proceed to the integrate process. So the integrate process actually delivered a mixture of large and small particles. And we conduct 2 experiments, and one is we conduct one more experiment, which is the control experiment, condition M, which is conducted at the same condition as condition A, but without adding anything. So for the larger particles obtained under condition A, the size and morphology were consistent with the crystal that we obtained from the standalone crystallization. Therefore, this suggests the spirit giant did not substantially alter the crystal properties as expected. And the small particles that were obtained from both condition A and M, they have a size of around 1 micrometer, appear amorphous. These are all characteristics of spray drying from a solution. And therefore, these particles were ascribed to the non-volatile compounds, and this can be possibly removed by a membrane process in future work. So next, we can look at the process yield and the insulin mass fraction. The process yield is defined as the ratio of the mass of insulin in the collected powder, the mass of insulin in the feed solution in the very beginning. So this means how much insulin we can recover from the entire integrated process. While the insulin mass fraction is the mass percentage of insulin in the spray dried powder. So we can see this figure. Actually, if you look at this figure, there's no substantial difference in both process yield and insulin mass fraction for all the tested conditions. For the process yield, it's in the range of 20 to around 34%, which is low compared to the recovery of the standalone crystallization. And we think because of the loss of particles during the cyclone in the spray dryer, because this spray dryer is not intended for collecting these small particles in this size range. So this can be further improved if we modify the cyclone or we optimize the spray drying conditions. For the insulin mass fraction, it is slightly over 20%. This also confirmed there's a large amount of other compounds in the spray dried powder. So finally, it's about the aerosol performance, and it is characterized by next generation impactor, which can simulate the dispersion of a dry powder from the inhaler to the lungs. And 4 parameters are used to characterize the performance, which is emitted fraction, which is the fraction of insulin that was emitted from the inhaler, and the fine part fraction, the mass fraction of insulin in the optimal size range, and also the mass median aerodynamic diameter and the geometric standard deviation, which is used to describe the particle size, aerodynamic particle size. So for all the test conditions that have emitted fraction of around 90%, this is the good dispersibility of the powder. While the FPF is in the range of 20 to 36%. which are generally considered acceptable for pulmonary drugs. But we observed a large variation for condition E and K, and for reasons that we don't know. For MMA, they're around or less than 5 micrometer, and GSD is in the same magnitude of as MMAD. So this suggests the CSD is relatively wide. But in general, the aerosol performance of the insulin crystals obtained from the integrated process has a promising performance, and condition L is considered optimal due to the good reproducibility and a high FEF. So now is the conclusions and outlook. So first, we developed a continuous insulin crystallization process, which allow for a recovery that is over 90%, with a size close to the optimal size range for pulmonary delivery. The second, the dried crystals that was obtained from the integrated process showed a promising aerosol performance. For future work, we might focus on the separation of the non-volatile compounds for spray drying, which can possibly be achieved by using a membrane process. Or we can investigate the potential benefits of the insulin crystals compared to their amorphous counterpart. And also we can improve the collection efficiency of the spray dryer by using, for example, a nano spray dryer. So I would like to thank my group mates, the technician from my university, and also Dr. Alan Chang from Talent Solutions for providing the insulin to us to conduct the pilot experiments. And thank you for your attention. This is the campus of USD.
Thank you very much for your presentation. So now the first question. What happens if you don't put nitrogen, without nitrogen slugs?
Without nitrogen, then we might, So the benefits of the segmented flow is that each slug can serve as a mini batch, and then the crystals are confined in the small space. So without nitrogen, we might achieve wider crystal size diffusion, but we didn't try that actually.
Okay, and so why shear is so important in insulin crystallization?
Many papers have addressed the effect of shear on the stability and the crystallization kinetics of proteins. For example, in one paper, they mentioned that insulin crystallization under a certain level of shear is likely to trigger the degradation of insulin molecules after some time. So under a very shear rate, we might denature the insulin.
Okay, and so you have a continuous process. So clogging is can be an issue. Did you notice any clogging in the microreactor at any condition?
Actually, we conducted some preliminary experiments. Sorry, go ahead. We conduct some. Actually, we conducted some preliminary experiments, which is operated as a very low flow rate. And in those cases, we did observe the clogging and plugging of this system. But later, then we increased the flow rate. And for the tested conditions in this operating range, in a liquid and gas flow rate of 0.6 or 1.8 milliliter per minute, we did not see clogging or plugging of the system.
Okay, can you go to slide 10? And just explain again why the residence time influences the crystal size distribution.
Sorry, can you read the...
So in slide 10, why the residence time influences the crystal size distribution?
Oh, okay, so here is actually not about the residence time. It's related, but the reason is not directly related to the residence time. So because of we guess the reason for the shift in the CSD is since some small particles can travel between slugs, and these particles have a larger residence time, and is able to travel to the other slugs And therefore the system, it takes some time for system to reach the proximate steady state in terms of CST. And also some particles, as a consequence, will have a longer residence time due to the traveling between the slugs.
Okay. Is your process compatible with the fact that protein are very expensive? And so if you go to a continuous process, is it sustainable, or what is the influence of the product price on your
yes,
design?
that's a very good question. Actually, yes, insulin is quite expensive, and so for each run we only operate for like normally we operate for round. For the integrated process, we operate for 1 hour, and for standalone crystallization, we operate for half an hour. And the initial concentration is around 2.4 milligram per milliliter. So for each experiment, it can cost I think it's around hundreds of Hong Kong dollars. So it's quite expensive, and therefore, but since we're actually the productivity of this process is not very high because we are using a tubing that has a very small inner diameter. So the mass of insulin that we can produce per hour is it's actually limited.
Okay, so thank you very much for your presentation. We're moving on the last speakers, and so I have to introduce now Denise Ling NG. She's a research engineer and PhD student at the National University of Singapore. Are you ready, Denise?
Thank you, Professor Beatrice, for your kind introduction. Good afternoon, everyone. Thank you for coming to today's talk. I'll be talking about my work on engineering spherical crystalline drug-colloid composites via microfluidic emulsion-based processing. So here I'm actually from a student from the Department of Chemical and Biomolecular Engineering. So here we'll start with an overview of my talk today. So firstly, it will start with the context for our discussion, which is pharmaceutical crystallization. Then after, I'll briefly touch upon microfluidic emulsion-based crystallization and how we can use that to form spherical agglomerates of neat drug particles, as well as co-formulate API excipients into a single spherical entity. Then after that, we'll talk about emulsion-based crystallization of API in the presence of colloids. And we'll show this for a hydrophilic drug, lamivudine, as well as 2 hydrophobic drugs, carbotegravir and naproxen. So here we'll start off with a brief overview of pharmaceutical crystallization. So crystallization is an important separation and purification process in the pharmaceutical industry because more than 90% of the drugs are actually delivered as crystalline form. This process also controls critical quality attributes for drug delivery as well as processing ease. So these critical quality attributes include the polymorphic form, the crystal dimensions, and the crystal morphology. In particular, for the crystal morphology, it can also influence processing ease severely. So for example, in the case of acicular crystals or needle-like crystals, this can affect the pneumatic transport of such crystals within different units in the plant. And crystallization is conventionally done in large stirred batch tanks in the pharmaceutical industry, which often have poor heat and mass transfer properties, and this results in poor control over the crystal size distribution. So one technique that my professor has been working on is the usage of microfluidics and emulsions to generate spherical mono-dispersed drug particles. And here, what I have is a video of our microfluidic emulsion-based, evaporative emulsion-based crystallization process. It's comprised of 2 steps. So in the first step is emulsion generation. So here what I have is a model hydrophilic drug, glycine, which is dissolved in water. Then after that, we emulsify them into mono-dispersed glycine-loaded emulsions. And this results in the dispensation of uniform contents into each droplet. And then after that, what we do is to collect these emulsions into a thin film, and we can control the film thickness to control the solvent removal parameters. And as the droplet undergoes evaporation, the droplet shrinks, the drug within it begins to supersaturate and crystallize into mono-dispersed spherical agglomerates. Yeah. So however, not in all cases, it will always crystallize into nice spherical agglomerates like we have seen for the case of glycine. For example, here I have the case of carbamazepine, where it actually crystallizes into bunches of needle-like crystals instead. And it does not give that nice spherical geometry, which is preferred because it has better flowability properties as compared to the acicular crystals, and it's so monodispersed in nature. So here what we can do is to actually incorporate a polymeric excipient. So here what we have done is to incorporate ethyl cellulose, a polymeric excipient, into it. And this actually helps to confine the crystals within a polymer matrix and results in the formation of monodispersed spherical agglomerates, spherical particles. Secondly, by controlling the film thickness of the drop in which the droplets are being incubated and solvent is being removed, we can actually control the rate at which solvent is being removed from the droplets. And here in the case of a monohydrophobic drug, in this case is commonly known as ROY for its red, orange, and yellow polymorphs. In cases where we have a thin film, the solvent removal rate is high, and this leads to the formation of a yellow polymorph. However, in other cases where we have a thick film and the solvent removal rate is much slower, we have an orange form. Furthermore, if we control the initial drug and excipient concentrations, we can also tune the overall particle structure, and this can result in a genus type of structure or a homogeneous type of structure. And this results in changes in the dissolution profiles as well. However, in some cases, by dissolving the drug and the polymer in the same entity, it may actually lead to the formation of amorphous solid dispersion. And this may pose physical stability issues such as polymorphic transformation over time. So here in the case of bristolfulvin and phenoldiphene, it actually crystallizes over time, and this may lead to changes in the dissolution and therefore the therapeutic efficacy. So besides the usage of polymers, there are actually many other types of excipients available, one of which is colloidal silica. So colloidal silica is a commonly used excipient, which is conventionally incorporated into solid dosage forms at the later stages to function as glidants or fillers. So here what we have shown here is that for a particle of ibuprofen, you can actually coat it with colloidal silica, and this results in a substantial improvement in the flow factor, which is a measure of the flowability of the powders. So what we would like to do here is actually not to use colloidal silica as a way to improve the flowability, but actually propose the use of colloidal silica to modulate critical quality attributes that we have mentioned earlier, such as the polymorphic form and crystal dimensions via emulsion-based processing. So we start off with a hydrophilic drug. So our model hydrophilic drug for this demonstration is lamivudine, which is a GSK commercialized hydrophilic antiretroviral drug. So in a typical experiment, what we do is that we dissolve lamivudine at a concentration of 80 mg per ml in water, and we disperse the colloidal silica into the same dispersed phase. It is then emulsified into monodispersed droplets using a continuous immiscible silicon oil phase. And these monodispersed droplets are collected into a thin film of 1 mm of the continuous phase, in this case is the silicon oil. Then we apply heat at 65 degrees to evaporate the water, and this actually results in the droplet shrinking. This results in the droplets undergo shrinkage. Then after that, crystallization propagates from one droplet periphery across the entire droplet. So at the end of the process, what we do is that we actually wash our particles to remove any traces of the surfactant. Then we dry it under vacuum and subject it to further characterization. So first and foremost, we characterize the changes in the lamivudine crystals morphology with and without colloidal silica presence. In the case of lamivudine particles, we see that the particles are roughly spherical in shape, and the crystals are lamellar-like. Whereas in the case of the presence of colloidal silica, it forms actually nice spherical particles. And these spherical particles consist of lamivudine crystals, which are fibrillar in nature, and they're all radially aligned to the droplet periphery. Next, to quantify the changes in the morphology, we took top-down FESCM images, where we characterize the cross-sectional dimensions of the lamivudine crystals in the X as well as the Y dimension. And we observed that in the absence of colloidal silica, the distribution of the crystals in the X and the Y dimension are significantly larger and wider in distribution. Then we also characterize the polymorphic outcomes of lamivudine in such particles via PXRD as well as DSC. In the case of neat lamivudine, we observe form 1, which is a 0.2 hydrate form, whereas in the presence of colloidal silica, we actually observe form 5, which is actually an anhydrous form. And this is actually in line with observations where nano confinement of crystallization under nano confinement in hard templates can actually lead to a formation of a different form. Next, to study the effect of the various types of colloidal silica properties on, or the colloid properties on the effects of the crystal attributes, we varied our colloidal silica sizes. So here we varied our colloidal silica size between 165 nanometers to 450 nanometers. And we observed that as we increase the colloidal silica sizes from 165 nanometers to 450 nanometers, we observe a corresponding increase in the crystal dimensions for the X dimension from 140 to 210 nanometers, and for the Y dimension from 230 nanometers to 400 nanometers. And thus we can conclude that the crystal dimensions can be correlated to the colloidal silica sizes at a fixed concentration of 40 mg per ml. And this level of control, to our best of knowledge, has not been reported in other work. So next, to determine if the surface chemistry of the colloid actually played an impact in the crystallization process, we then chose to keep our concentration constant and vary the type of colloid that we are using. So here for interest sakes, we are using colloidal polystyrene sulfonate in our process. And we observed that the composites which were formed at the end are also spherical in nature. And in particular, we observed that the outcome when it comes to the lamivudine crystals, they are also fibrillar-like and radially aligned with regards to the droplet periphery. The crystal size, the crystal cross-sectional dimensions in the X and the Y direction are also significantly smaller and narrower in distribution. And the polymorphic form observed in such composites are also form 5, which is identical to those observed in the colloidal silica case. Thus, this suggests that the process may not be influenced by the colloid surface chemistry. However, it is not sufficient to just apply it to the hydrophilic case, because that limits us to a very limited number of drugs, and a lot of the drugs are actually hydrophobic in nature. So what we like to do is to generalize our technique so that we can apply it to a wider range of drugs, and in this case, hydrophobic drugs. So here to generalize our technique further, we try to implement it for a hydrophobic drug, but it comes with its own set of challenges. So here we have to implement the organic solvent as our dispersed phase solvent instead of using water as we did in the case of lamivudine. And with this, it is difficult to disperse a colloidal silica in an organic solvent. So here I have an image of trying to disperse colloidal silica in dichloromethane, one of the commonly used organic solvents in oil and water emulsions. And secondly, the other issue is that we need to prevent the colloidal silica from partitioning out into the continuous phase, as you can see over here. Otherwise, the colloidal silica will not be able to exert its effect during the crystallization process. So to overcome this, we propose two solutions. So firstly, we surface modify our colloidal silica using silane chemistry with 3-amino propyl trihydroxysilane. And we disperse and confine them in a co-solvent mixture of dichloromethane and benzyl alcohol. And here we can see that we are able to do that quite successfully. The droplets here actually appear transparent because of the matching of the refractive indices between the colloidal silica as well as the co-solvent mixture. Then we perform the same evaporative emulsion-based processing for using a model hydrophobic drug, which in this case is cabotegravir, a hydrophobic antiretroviral drug. So in the case of both neat case and so as the composite case, we observe that both of them form nice spherical particles. And in particular, with the neat case, we observe that it forms large left-like crystals. And whereas in the case of the composite, we see that it forms very thin left-like crystals. And we also quantified the changes in terms of the crystal width. And here in the case of cabotegravir, we observed that it forms a distribution of 429 nanometers plus minus 116. But in the presence of colloidal silica, we see a significant narrowing and a significant decrease in the crystal width size. We then proceeded to look at the polymorphic outcome, and in both cases, we observed that both the neat cabotegravir as well as the composite case, both of them are of the same polymorphic form, which is form C1. And with this, we can try to implement dissolution to study the dissolution, as any changes in the dissolution can be directly correlated to the differences in the crystal dimensions. And here, in the case of carbonatogravure, we observed that actually in the case of our composites, it can dissolve to a much greater extent as compared to the neat case. And this suggests potential in implementing it to address the dissolution extent of hydrophobic drugs. Next, we also note that the evaporative emulsion-based technique may not be applicable to heat-labile drugs, especially in cases where the dispersed phase is a non-volatile solvent, such as water, where heat is needed to actually accelerate the process. Hence, we also develop an extractive emulsion-based processing technique. So here, what I have done is to use benzyl alcohol water as our emulsion system. And here, naproxen is used as our model hydrophobic drug. And likewise, colloidal silica was also surface modified using APTS. And in this case, naproxen and colloidal silica is then emulsified into monodispersed droplets using a microfluidic device again with benzyl alcohol saturated water. So this helps to prevent any extraction from occurring during this process in the tube. And then it only occurs within these thin film conditions at the outlet here. So this helps to prevent any clogging within the tubes. So what happens after the droplets have been collected into this thin film is that the benzyl alcohol droplet is benzyl alcohol is being extracted into the film, and this results in droplet shrinkage, and therefore the naproxen in it begins to supersaturate and crystallize. In the case of neat particles, we observe that it forms spherical particles with large left-like crystals, and the crystal widths likewise are approximately 1 micron in size. Whereas what's interesting is that in the case of our colloidal silica composites is that it forms a colloid poor region where large left-like crystals approximately about 1 micrometer in crystal width are formed. And on the colloid rich region, we actually observe a significant narrowing and narrower distribution of crystal widths as well as smaller crystal width sizes. Next, we also varied temperature to see if it will influence the crystal outcomes. So here in the case of the 20 degrees, which as we saw earlier, it formed a bi-domain. When we increase the temperature, it also likewise formed a bi-domain particle as well. However, what's interesting is that across the two temperatures, at the lower temperature of 20 degrees, it forms a left-like crystal habit, whereas in the case of a higher temperature, it forms a prismatic crystal habit. And on the colloid-rich region, a significant difference was also observed. So here we have thin left-like crystals that are dispersed among colloidal silica, whereas here we have prismatic crystals that have colloidal silica actually being engulfed inside the crystals itself. So with this, we have briefly demonstrated that we are able to form hydrophilic drug-colloid composites in the case of lamivudine via evaporative emulsion-based processing. And we observed that actually we can obtain a different polymorph in the presence of colloids. And we can actually tune the crystal cross-sectional dimensions just by changing the colloidal silica sizes. And the similarity in outcomes between the colloidal silica and colloidal polystyrene composites suggests that this process may actually be surface agnostic. We then move on to extend and generalize our technique for hydrophobic drugs. And we demonstrate it for both evaporative and extractive emulsion-based processing, where we are also able to actually tune the crystal dimensions, crystal habits, and particle structure. Lastly, ongoing work includes understanding the formation mechanism and also further study the correlation between dissolution and particle structure. And this work would not have been possible without the support and the advice of the following people, as well as the generous funding from the following funders. Thank you.
Thank you very much for your presentation. Now we are going to the questions. So can you tell us about some words about the reproducibility of your results? Are you sure that, because you have difficult characterization with effects on counts, of course, which are sometimes small. So are you sure that these effects are reproducible?
So here what we have done is actually we do it for ensemble of particles, so not only one, so across probably around 50 or more particles. And after that, what we do is that we take images of the top-down, for example, in the characterization of the crystal cross-sectional dimensions. We actually take top-down FTSM images and we do image analysis across 300 crystals. So I think it is fairly reproducible.
Okay, so another question is, have you an idea of the influence of the size of the droplet at the beginning? Because you are creating supersaturation by evaporation. So the interface is very important.
Yeah.
And so do you think that the size of the droplets is linked to the homogeneity of your solution inside?
So microfluidics itself, actually by the physics of microfluidics, it actually enables firstly the contents of each droplet to be actually uniform and the droplet size to be very uniform, plus minus 10% standard deviation. So that is actually really like quite well controlled. And plus we also control the film thickness, which is the rate at which our droplets, the rate at which the solvent is being removed from the droplets. So I think we have tried to actually control on all fronts that we have known about the process.
Okay, so you have tried large droplets, small droplets?
Yeah.
And so you have the same results?
Yes, we have.
Okay. Okay. So you have very, very nice particles, spherical particles. And what is the proportion of colloidal silica in your particle? And this proportion is acceptable for pharmaceutical application?
So in the case of colloidal silica, so it actually has a fairly high tolerability in the human body. And for the cases that I've studied, so I've looked at the dosage amounts that are typically like administered in the case of lamivudine, for example. And actually the amount of colloidal silica that if we were to administer the same amount of dosage of lamivudine is actually below the tolerable amount that is reported in the literature.
Okay. So thank you very much for your presentation. Thank you very much to all the speakers and this very interesting discussion. Now I give I come back to Marco Mazzotti for the conclusion. Thank you.
Yes, thank you very much, Beatrice, for chairing this session. I think we had a great start, and I would like to, after thanking Marta, Jonathan, Ruipeng, and Denise for the very interesting talks, I would like to thank also the participants and invite all of you to attend the second and the third days of crystallization webinars tomorrow at the same time and on Friday at the same time. And you see on my screen the program. Tomorrow we will focus on fundamentals of crystallization and on Friday on industrial applications. So I wish all of you a good day and a good evening, and look forward to seeing you tomorrow and the day after. Goodbye.